Good day, ladies. So now we're going to continue with our process of mitosis. If you remember from the previous the previous video, we spoke about the importance of mitosis and how useful it is. It's allowing you for repair, replacement, growth, and asexual reproduction. So just in case you forgot about that. All right. So now the first phase is prophase. And I want you to remember IPMAT, IPMAT, and right at the top of the cross of your page. And that will remind you of the sequence of all the different phases. So inter, pro, meta, ana, telo. All right, and then you just put phase at the end of each one of those. So the first one we're going to do is prophase. It normally gets broken up into two parts, early prophase and late prophase. But long story short, remember we spoke about the centrioles yesterday, which I said where spindle fibers will form from. So one centrosome creates these centrioles, and they move to the two different sides of the cell. However, these are not found in plants, and the ones that are found in plants are called MTOCs, or microtubule organizing centers, but you don't need to know that part. You just need to know that animals have centrioles. Then from these centrioles, and you can see that I've actually put in the little diagram over there of the centrioles, and off of that, they're speaking about the microtubule triplets. I don't want to know about that. But those little things can actually create the spindle fibers, which we'll see from the diagram at the bottom over there. So the spindle fibers form and attach to the centromeres. So remember, those are the proteins that are holding the two sister chromatids together, and the nuclear envelope breaks down and the nucleolus disappears, because now we need direct access to all of those chromosomes inside of the nucleus. So I want you to please draw this. So we have early prophase over here and late prophase. Early prophase, you can see that the nuclear envelope is disappearing, or the nuclear membrane. Spindle fibers are being produced, and then in late prophase, you can see that the spindle fibers have actually joined to the centromeres of the chromosome. All right. Then we go into metaphase. This is the shortest phase. This is where the centrioles travel to the poles of the cell and anchor to the spindle fibers. So you'll see that they're also talking about the anchor aster fibers over there on the diagram. And I don't necessarily need you to know what those are, but that is literally just where all these spindle fibers are coming from. So it says the condensed chromosome line up. On the equator, the spindle fibers link to the chromatids of each chromosome to the opposite poles. Okay, so on the left-hand side again, it says chromosomes move to the metaphase plate, or you could just use the word equator. I am 100% okay with you just calling it the equator of the cell, and that's the center of the cell. And it's aligning it on the center as well, so that it has equal access to each and every single one of those chromosomes, so that it can now split it apart. So this is your diagram for metaphase. Please notice it has aster fibers, centrioles, spindle fibers, and then I also want you to include the label of chromosomes, okay? Then we have anaphase. This is my favorite phase just because it's the one that everyone remembers for some reason. So where it says number 3, as the spindle fibers shorten, the chromatids are pulled towards the poles of the cell. Okay, let's go to the left. Centromeres separate and spindle fibers contract. So these little protein spindle fibers can actually start pulling on each other. And when they pull, they actually split that chromosome directly down the middle and then pulls the sister chromatids to either side. Okay, so the sister chromatids separate and they move apart towards the poles. After separation, the chromatids are now considered chromosomes. So remember I said to you that when they are joined, they're sister chromatids and they make up a chromosome. When they are individual, they are also known as chromosomes. Okay, so when they start being pulled to either side, and please note the V-like shape that the chromosomes or sister chromatids actually start producing, okay? And that indicates to you that anaphase is now happening. So here's your diagram for anaphase. Please, when you draw this, please make sure that the arms of the chromosomes create that very V-like structure, okay? So they're no longer sister chromatids, they're now chromosomes, and they're pulled to the poles. And what created these poles again? The centrioles, which are made, or like the centrioles and two centrioles, and it makes a centrosome. Alrighty, so then we finally have telophase. Telophase is where the nuclear membrane and the nucleolus reappear. The chromosome uncoils to chromatin, and in the end, two genetically identical nuclei are present. Okay, so this is mainly for plants. Okay, and we'll talk about how, sorry, animals, and we'll talk about how plants change ever so slightly. And you can see that they're starting to almost like pinch off in the middle as that new nucleus is starting to form around them. So the cleavage furrow develops in animal cells, and a cell plate develops in plants. So now a cell plate is a new cell wall that starts forming in the middle of the two new developed cells. And those two new developed cells actually need to be separated by a cell wall, because remember, a cell wall itself can't constrict, because if it constricts, then the whole thing is going to snap. So this is your second last drawing. This is cytokinesis. So this is the actual pulling together of that furrow. And you can see on the left-hand side that slightly yellow, orangey brown picture over there. That is cells actually starting to pinch off, okay? And there's a nice micrograph at the top left-hand corner over there. So please draw this. And finally, we have cytoplasmic division. So we had a nuclear division, which was the splitting of the nucleus and the DNA, and now we have cytoplasmic division, okay? Also known as cytokinesis. So the cell plate becomes complete in plants, so you can see on the right-hand side in there, and in the end, two separate daughter cells produced with singular nucleus. So please make note that when you go back and you have a look at all of this, that you notice that the DNA doubled. It's like copied versions of each other joined to each other, then went through the whole process. Those copies then split off of each other, giving a set to either to each one of the cells, and eventually the whole cell membrane just pinches off and two cells then produce. Whereas in plant cells, that cell plate forms in the middle, and that's what allows the plant cell to. Here is name the phase in the micrograph. So at the top left-hand corner, we can see the chromosome formation. And also to a large extent, it's, we've lost the nuclear membrane, so that's more prophase. Then below that, we've got all the chromosomes laying on the equator. That's metaphase. On the top right-hand corner, we have that V-like structure with all the chromosomes or sister chromatids, and those are that phase is anaphase, and directly below that is telophase as a whole. Okay, you don't even have to talk about early and late or nuclear division and cytoplasmic division. You just talk about telophase as a whole. All right, to finish this off, I've included a cool little GIF over there where you can see some animal cells actually going through the process of mitosis. So they just stained it with fluorescent dyes, and then they shine a light over it and actually changes color, and we can actually follow what's going on. But now I would also like you to do learning activity 2 on page 126 and 127. All right, ladies, I know it seems very quick, but when you start writing all of it down and drawing, it should take you some time. Please also make sure that you do this 100% properly, and if you have any questions, please ask, because this is so important and it directly relates to what you do in matric when we do meiosis. All right, have a good day, ladies, and I'll speak to you soon.